genetic screening, what it is, the different types, and how to apply it to specific scenarios. Made by Vanessa, Patum, Summer, Ashley, and Karolina.
What is genetic screening? Genetic screening is a technique used by scientists today to identify and characterize genes involved in various biological processes. Through the study of genes, scientists can obtain insight into how these genes affect different functions and the overall health of many organisms. The process of genetic screening begins when exposing a given population to a mutagen that alters the individual's DNA sequence. Once the population has reproduced, scientists observe the offspring, otherwise known as the progeny, to detect any defects or changes in the traits being studied. Many induced mutant genes will be recessive. There are two ways that gene identification is brought upon in genetic screening, including forward genetic screening and reverse genetic screening.
Starting with forward genetic screening, this process is used to identify genes responsible for a mutated phenotype by inducing random mutations in a large population. Once the population has reproduced, scientists seek out non-specific phenotypes that could be due to the disruption of the biological pathway. In comparison, reverse genetic screening begins by creating a mutation in a specific gene, allowing reproduction to produce new offspring and observing the phenotypic consequences of the distinct mutant gene.
Now that we have learned more about what genetic screening is, let's focus on our experimental question. A frog breeder wants a more diverse range of African clawed frogs for an upcoming reptile exposition. How could the frog breeder use genetic screening to achieve diversity in his frog collection? The breeder exposed 2 of his 4 frogs to ultraviolet light in order to introduce mutations, and then he mates the 2 mutated frogs, resulting in 4 offspring. We can assume based on the observed traits that the ultraviolet light did in fact cause mutations in the African clawed frogs, therefore resulting in a diverse range of phenotypes. Before we continue to further analyze the process, let's quickly discuss the African clawed frog. African clawed frogs, scientific name Xenopus laevis, are a non-poisonous aquatic frog species native to Africa. They are also an invasive species which can be found around Europe and Southern and Northern America. They have webbed feet fit with sets of 3 clawed toes. African clawed frogs live in shallow waters with an optimal environmental pH of around 7.
In the first step, you should select and prepare parent frogs. You should select 4 frogs and split them into 2 groups. 2 would be the control group, unexposed to UV light, while the other 2 in the experimental group will undergo UV exposure. The parent frogs have different traits. Spotted frogs with long claws will show dominant features, while unmarked frogs with short claws will show recessive traits. In step 2, you should induce mutations using UV light. You should expose the experimental group to UV light for a set period of time. UV radiation is known to cause genetic changes. Targeting germ cells should ensure mutations are inherited, simulating how environmental factors influence genetic variation. In the next step, you should breed to identify mutant traits. After UV exposure, he should breed the experimental frogs. Their offspring can then be compared to the control group to check for new mutations. In the next step, you should observe and analyze offspring. You should examine the offspring for changes such as lighter pigmentation, claw length between long and short, shorter webbing, possibly affecting swimming of the frogs, a shift in the pH tolerance. In the next step, you should confirm the traits with pedigree analysis. To confirm the changes are from mutations, you should conduct a pedigree analysis. Tracking traits across generations should reveal if they follow normal inheritance patterns or they result from random mutations. And finally, you should collect and organize the data. You should organize the results in a table. Before UV exposure, the parental frogs had predictable traits. After breeding, new traits appeared, supporting the idea that UV radiation can change genetic expression.
After mating the 2 frogs subject to UV, the results of the forward genetic screen of their offspring show that multiple mutations have occurred. The resulting novel traits included various physiological changes. The mother frog possesses the dominant traits, which are a spotted back and long claws, while the male frog possesses the recessive traits, being an unmarked back with short claws. All the offspring possess new traits different to those that either parents have possessed. The forward genetic screen revealed various new traits, the first being that instead of being olive green like the parent frogs, the progeny were either light tan or albino. The frogs also had claws that weren't short or long. This means that their claws did not benefit their survival greatly because they were not able to grasp things well with long claws or stick to surfaces with short claws. Another phenotypic trait observed in the progeny was that the original webbing on the parents reached the start of their claws. However, in the progeny, the webbing was much shorter and caused them to move much slower in the water than the parents had. The frogs also displayed variation in the preferred acidity of their habitat. The frogs became sick in the same habitat as their parents, which was settled at a pH of 7. However, the offspring did not seem to get better until they were put into a new habitat, adjusted to a pH of 8.5. To ensure that the traits were indeed true mutants, we observed the traits through pedigrees of all previous frog generations the breeder had owned. Because all the mutant traits produced are recessive, it would be impossible for the entire generation to each possess and express these recessive traits, since only half could be homozygous recessive. Considering that the traits the offspring possess, such as the trait for albinism and light-colored skin, do not even exist in the parents or the grandparents, supports the fact that they are completely novel traits. This procedure confirmed that the traits were in fact novel traits, resulting from the mutation done in the parents.
Now, here's some key takeaways from genetic screening. Genetic screening is largely important as it helps to analyze and identify genes. This is applicable to scientific research, medical and forensic research, and for biodiversity in general. As previously mentioned, there are two main types of genetic screening, forward and reverse. Forward genetic screening identifies genes based on observed mutations. This is what we use to achieve diversity among the African clawed frog population, using ultraviolet light to induce mutations, whereas reverse genetic screening examines the effects of specific gene modifications. The observed mutations on the frogs following exposure to the radiation were a change in environmental pH preferences, skin coloration, level of webbing, and the length of claws. These are all novel traits which emerged from the mutations. Ultimately, the answer to our original question is that the frog breeder was successful in using forward genetic screening to achieve diversity within his collection of frogs using ultraviolet light, inducing mutations that resulted in novel traits. Thank you for watching and listening to our presentation on genetic screening and its role in studying mutations, identifying genes, and genetic diversity.